Okay, hi everyone. My name's Dan. I'm a postdoc here at McMaster, and I'm really excited to have another NeuroMusic this year. It's great to be with everyone. Good morning, afternoon, evening, wherever you are. We have people joining us from all over the world. So I'm really happy to introduce our first speaker, but first I just want to remind everyone that after each of the talk sessions, there's a Q&A, and we want everyone to engage and ask your questions. If you're going to ask a question, please post it using the Q&A function in Zoom, not using the chat. And also, I think it's one thing we can take advantage of by doing things virtually is that you can post questions during the talk. So if something strikes you during one of the talks you're watching, you can post it in the Q&A right then. And then we'll get to the Q&A, the questions after each talk. Okay, so I'm very, very happy to introduce Dr. Stefanie Hoehl. Stefanie is the head of the Research Unit of Developmental Psychology at the University of Vienna and leads the Wiener Kinderstudien. Stefanie completed her undergraduate studies in psychology at Heidelberg and did her PhD at the University of Leipzig in 2008. And she was back in Heidelberg to do her habilitation in 2013. Then from 2016 to 2019, she had a prestigious group leader position at the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig. Stefanie and her group are very productive and impactful. She has over 2,500 citations, and just this year, she's published numerous papers using a diversity of methods, including EEG, FNIRS, hyperscanning, eye tracking, and more, and all to study social and cognitive development in early childhood. So today, Stefanie's talk is titled Getting Attuned to Others, Social Synchrony in Early Human Development. I know we're all eager to hear her talk and discuss her work in the Q&A afterwards, so let's get to it. Here is Stefanie Hoehl.
Hi, I'm Stefanie Hoehl, and I'm a developmental psychologist at the University of Vienna. Very happy to present our research today on getting attuned, neurosynchrony in caregiver-child interactions. Before diving into our empirical work, I'm going to give you a brief theoretical background on why we are interested in interpersonal rhythms and synchrony in early development. I'm then going to present a series of studies where we looked at caregiver-child interactions with preschoolers and their parents. And in the last part of my talk, I'm going to present a study where we were interested in mother-infant interaction, and there especially in the role of physical proximity and touch. Yeah, given that you're interested in music and rhythms, I probably don't have to go into much detail into why rhythms are interesting and relevant for social interaction, but I'm going to give you some insights or some glimpses of information on rhythms in caregiver-child interactions, especially. So if we look at characteristic caregiver-child and caregiver-infant interactions, rhythms are really ubiquitous, rhythms abound. There's particular rhythmicity to infant-directed speech. There are proto-conversations, this back and forth, even with pre-verbal infants, that kind of has a form of a conversation, but is pre-verbal, has a particular rhythmicity. And then caregivers, of course, use rhymes, nursery rhymes, music, and singing. In addition, affective touch is characterized by particular rhythms. And of course, infants experience interactional rhythms when being carried or rocked, or in interactive games with their caregivers. And on a larger time scale, there are also rhythms in terms of daily routines that infants experience and children experience with their caregivers. Now, all of these rhythms are probably not there by chance. They probably serve some functions, and two aspects that I think are important to consider here is that rhythms make the world more predictable for the infant, who initially, of course, after being born, lacks control over what is happening most of the time, but at least these rhythms make the world predictable, and it's easier to compute what probably comes next. And also they help caregiver and infant achieve interpersonal synchrony. And we have talked about these ideas and laid them out a bit in some recent perspective papers. Now, there is actually already quite a lot of research on behavioral and physiological synchrony and caregiver-infant interactions, notably seminal work by Ruth Feldman and her group, who looked at behavioral synchrony on different levels here. In the picture, you see affective expression synchrony, but also physiological synchrony in different forms, like heart rate coupling or endocrine fit in this particular graphic, oxytocin release. And they showed that behavioral synchrony and physiological synchrony are closely related. They go hand in hand. They're probably foundational for the infant-parent bond and for the formation of a secure attachment. And also, again, as the infant lacks at the beginning critical self-regulation and affective and also emotional regulation capacities, the synchrony is probably critical for the maintenance of homeostasis in the infant, for instance, in terms of stress regulation, and also for the formation of infants' models of the self and others in close relationships, as nicely laid out in the paper by Fotopoulou and Tsakiris on the bottom of the slide. Now, there is still a lot of open questions, however. As I've mentioned, all of this research is focused very much on the affective domain and attachment relationship formation, but we know much less about the role of interpersonal synchrony for social learning, for communication and cognitive development. And also we know much less about neurosynchrony in early development. Now, why is neurosynchrony in particular of interest? Well, this research is very much inspired by research into speech processing from adults or with adults, where neurophase coupling has been ascribed a functional role in the support of communication, of information transfer between two individuals. Now, if you look at brain oscillations, what they essentially are fluctuations in the excitability of the underlying neuronal populations, and the very same physical stimulus can be processed more or less efficiently depending on when in the cycle of a neural oscillation it occurs. Yeah, so this is of course a very simplified illustration from a paper by Peele and Davis, where they showed that the same physical stimulus can occur at a time when processing efficiency is high or low, and this of course affects whether it's detected, whether it's encoded, to what degree it's processed. Now, if I think about a rhythmic stimulus or a quasi-rhythmic stimulus like speech, also singing, music, of course, we can consider a situation with suboptimal phase alignment where this rhythmic stimulus always occurs kind of when it's not processed efficiently, or we can consider optimal phase alignment, which would then kind of boost processing. It would amplify basically the signal in the brain and would therefore support processing, and in the case of speech rhythms, support communication. So thinking about how interpersonal synchrony might come about, if we're thinking about a diet that's actually interacting reciprocally, communicative rhythms probably play a role as depicted here on the picture on the left. Speaker-listener neural coupling would then be transmitted or occur through the rhythmic speech stimulus. Yeah, we have speech production in the speaker, the speech signal that travels through air to the listener, and then hopefully, in the ideal case, a resonant frequency in the listener's brain. But other signals could do the trick as well, such as biological motion, and as I mentioned, also affective touch. Yeah. So in this case, neurosynchrony would occur within the diet, basically. But of course, also environmental stimuli and rhythms might play a role in this process. Here, for instance, focusing on something together at the same time, call that joint attention, but also external rhythms like listening to music together. So we can also think of situations where the synchrony not only occurs within the diet, like by transmitted or supported by signals within the diet, but also by focusing jointly on something in the external environment. So that would be a triadic situation. And of course, these processes very likely go hand in hand in a lot of cases. They're not mutually exclusive. So if we want to actually research these interesting phenomena and processes in early development, we have to move towards different paradigms than classically. So classically, we've tried to create very controlled experimental situations in the lab, the infant looking at something on the screen with little movement, but also with very limited ecological validity. And we've step by step moved towards kind of more ecologically valid situations where here in the study, an adult was engaging with the infant in a live interaction to study joint attention. And now more recently, we're starting to look at the brain activity of the adult interaction partner as well, to be able to capture the dynamics of social interactions, especially the back and forth, the mutual attunement and the reciprocity that goes on in these interactions. So to move to our first study that we did to look at neurosynchrony in live and dynamic caregiver-child interactions, our starting point was that interbrain synchrony was observed in both adult diets and also in parent-child diets in cooperation tasks that were very simple and also still highly controlled. So in these studies, for instance, the study by Vanessa Reindl, 2018 in NeuroImage, and Kerstin Konrad, they asked the participants to basically press a button in close temporal proximity, so as closely together in time as possible. And what they found was that when diets succeeded in this task and pressed the button approximately at the same time, this was then rewarded with a little animation. Those were the diets that also showed a higher degree of neurosynchronization as measured with functional FNIRS. Now, we wondered whether interpersonal neurosynchrony could also play a role in more naturalistic task context. So something that a parent and a child might actually experience in their daily lives, such as solving a puzzle task together. Why we were interested in that? Well, first of all, again, in order to achieve a higher level of ecological validity, but especially because we wanted to look at the interactional dynamics in this situation, because we figured that if we put caregivers and children in this situation with very little constraints, that we would find a lot of inter-individual or interdyadic variability. And that's something that is very interesting to us, to look at what interactional qualities are actually associated with higher or lower levels of interpersonal synchronization. So here we asked, does neurosynchrony between mothers and their preschool-aged children depend on the quality of the interaction and also maternal sensitivity? Because this is a variable that has been documented in attachment research to play a role for mutual attunement and the formation of a secure attachment. But then also in turn, does increased neurosynchrony predict more successful collaborative task performance in a complex and naturalistic task, such as solving a puzzle together? So what we did is we gave mothers and children some geometric wooden shapes, a time crumb game basically, and asked them to recreate templates. And this is what it looks like. So here you see the wooden shapes on the bottom and the template on the top on the table, and you see that both are trying to figure out how to recreate the template. They are starting to communicate about that. The task was to do this together. Little more instruction than that. So again, we aimed at getting a lot of variability on the behavioral level. And here you see them starting to coordinate and communicate. And those were, in this first study, mother-child diets with children aged 5 to 6. We were using functional near-infrared spectroscopy in this study, as this previous work also did before us, with a more simplified task, which has the advantage that it's not much restrictive in terms of the behavior that we can observe. We can use that in naturalistic tasks, as you saw. We don't need a huge scanner or anything, and it's not so much affected by movement as compared to EEG, for instance, but we can only assess brain activity on the cortex, and also only in some selected areas, because we cannot cover, at least with the machine that we have, the whole head. So what we focused on was, on the one hand, the temporoparietal junction, which has been associated with theory of mind and perspective taking. And then also the dorsolateral prefrontal cortex, which has been implicated in synchronization, neurosynchronization during cooperation compared to competition in these simplified tasks. Our main hypotheses were that mother-child neurosynchrony would be associated with maternal sensitivity, and also behavioral dyadic reciprocity as a marker of the interaction quality. And also, on the other hand, that neurosynchrony would be associated with more successful joint task performance. This was operationalized as a number of puzzles that the child and mother were able to solve together in a given time. Okay, so we looked at the FNIRS time series from both heads, from the mother and the child, and we used wavelet transform coherence as a measure of neurosynchronization. Again, this is something that, there's different measures that one can take, and we chose that mainly because it was used successfully in the previous research that we based our hypothesis on. And we looked at this coherence between mother and child in 3 different contexts. The one was the collaborative joint task performance that I just showed you in the video, but we also had an individual task where mother and child did the same puzzles, but they were separated by a little screen in between, so they were not interacting with each other in that condition. And then we also had a resting state condition where we just asked them to sit back and relax. And the first thing you see here is that indeed, as we would expect, neurosynchrony is increased during collaboration as compared to the other conditions. So when there was mutual engagement, there was a higher level of neurosynchronization. So this was reassuring. Still, we ran some control analysis. This is also something that's very common in this field. What we did here is we performed a random pair analysis where we compared our original pairs, so the mothers that were actually interacting with their children in the room, with randomized couplings. So we basically just took the time series of one mother and paired it up with the time series of different children who did not actually interact with each other. We just paired them up to see whether there was some kind of random synchronization, maybe something to do with the task or the machine or the room. And what was very, very reassuring in this analysis is that indeed in the original pairs, those that actually did collaborate, that did interact, we see a higher level of coherence as compared to the random pairs. And now we can look at the kind of interesting associations between neurosynchrony and the behavioral parameters. And we, as expected, found that higher neurosynchronization went along with higher cooperative success. So the number of templates that were solved by the diet was higher in those diets with a higher level of neurosynchronization. Maternal sensitivity, on the other hand, did not show any association, neither with the self-report, where we just ask mothers with a questionnaire how sensitive they judged themselves to be, neither with a video coding from the task, from the interaction, where we also looked at maternal sensitivity. However, I have to kind of point out that maternal sensitivity can, of course, also be assessed in a more detailed way in parents' homes for longer periods of time. So we cannot exclude completely the possibility that maternal sensitivity plays a role in this context, but not with the measures that we used in the study. Agency of the child, on the other hand, showed a positive correlation with the neurosynchrony. Higher child agency was associated with higher levels of neural synchronization, which points towards the role of the child actually in the interaction, in driving this synchronization. And as expected, again, behavioral reciprocity was positively associated. So here we looked at the turn-taking quality, the back and forth between mother and child, which was very nicely shown actually, or exemplified in the video I showed you, where both partners actually do respond contingently to each other. This was not the case in all of the diets, so we found a lot of variability in that, and we found that those diets with a higher level of behavioral reciprocity also showed a higher level of neurosynchronization. This is actually very much in line with a result we observed in a related study. Here we invited the same mother-child diets to engage in a free verbal conversation for 5 minutes. So again, a very free, very naturalistic context. And we coded turn-taking in this verbal turn-taking in this free conversation. Over the 5 minutes of conversation, we looked at 30-second epochs and coded the level of turn-taking, and also analyzed again neurosynchrony with wavelet transform coherence. And what we found was here that in those diets that over the 5 minutes had a high level of verbal turn-taking, neurosynchrony actually increased over the 5-minute conversation. In those diets that had a low level of turn-taking during the conversation, a low level of neural synchronization was maintained over the 5-minute conversation. Yeah, so again, we see that it's actually the diet, or the quality of the interaction in the diet, the reciprocity, the turn-taking, contingent responding, that seems to drive neurosynchrony, or at least seems to be associated with high level of neural synchronization. So to conclude, we saw this first research as a proof of concept. We do see mother-child diets show heightened neurosynchrony during naturalistic collaboration, also during free verbal conversation, as compared to these control conditions that we tested, like individual problem solving and rest. And we also found that higher neurosynchrony during the collaboration predicted more successful joint task performance in line with previous research. We think this speaks to neurosynchrony as a biomarker for mutual task engagement. We also found that higher behavior reciprocity and also higher levels of verbal turn-taking in the mother-child interaction are associated with greater neurosynchrony as well as child agency. So this points to the role of the active child in the interaction, and also dyadic reciprocity as an important factor. You might be wondering now, what about fathers? We also ran a study with father-child diets with children, again, aged between 5 and 6 years old, and we asked them to do the same task with a puzzle. And what we found, again, as also in the mother-child diets, is that there's a higher level of neurosynchronization between fathers and their children in the cooperation condition compared to the individual condition and compared to rest. So we can replicate this general pattern of results, and looking at what variables are associated with father-child neurosynchrony, we found in particular the self-reported role of the father played an important role. So the degree to which fathers identify with their role as a father, as a caregiver, that was associated positively with the level of neurosynchronization with their child. Okay, in the last part of my talk, I now would like to focus on a study on mother-infant interaction. So we're basically moving towards much younger children now. And as I've said, there is substantial research already on physiological synchrony, also behavioral synchrony, but a lot of open questions regarding neurosynchronization. So we've seen these links between behavioral synchrony and physiological synchrony, but we don't know as much about how neurosynchrony is established in the mother-infant diet, and also whether it's associated with behavioral or physiological synchrony. So what was very important for us in this study was to actually collect data and analyze data on all three of these different levels. So one question that we asked, and that comes up a lot in this line of research, is whether interpersonal neurosynchrony is mainly driven by sharing perceptual input, or whether it really actually has to do something with the interaction, with the quality of the interaction. Then we were interested in the role of close physical proximity, whether that affects neurosynchrony in infant caregiver diets, and also what qualities of the interaction in general are associated with higher neurosynchronization. In particular, again, we looked here at affectionate touch. And finally, since we're able to look here at the different levels together, we were wondering whether physiological and neurosynchrony are related. And there's very little research on this, some studies in adults, but very little research on this in development. So what we did is we invited 81 mother-infant diets to the lab when the infant was 4 to 6 months old. And then we visited them again when the infants were 12 months old, but I'm focusing today on the lab data. And here we assessed, again, these 3 levels, basically. We looked at brain activity again via functional near-infrared spectroscopy, but we collected at the same time respiratory sinus arrhythmia from mother and infant with electrocardiogram, and we, again, video recorded the interaction. And those were the settings that we used in the mother-infant study. Obviously, we cannot ask them to collaborate on a puzzle task or even engage in a verbal conversation. What we decided was to ask them to watch little aquarium videos together, one time in the proximal watching condition, which was basically the mom holding the infant on her lap. And this was compared to a distal watching condition where the same videos were shown on a tablet, but there was no touch. Yeah, the infant was seated in a seat. And then we also asked mothers to engage in a free play interaction with their infant. We asked them to play with the baby as they usually would, but we asked them not to use toys and not to sing, because we wanted to focus here on rhythms in the interaction that are not affected by musical interactions. Yeah. So this is just again a schematic of the task design: distal joint watching, proximal joint watching, and the face-to-face interaction. Again, we had to decide which regions of interest to look at in the brain. We here decided to go for frontal regions associated with the processing of affective touch, in particular the inferior frontal gyrus, and then also medial prefrontal cortex, because synchronization of brain activity during free play between older infants and their mothers was recorded in this area in a study by Lisa Piazza et al. And also in the social interaction studies between preschoolers and their moms. Okay, again, the results. Again, we looked at wavelet transform coherence in the diets, in those diets that retained data of adequate quality. And what we found here was, on the one hand, that coherence between infants and mothers was higher during free play compared to the proximal watching condition. So the face-to-face interaction was actually what induced the highest level of neurosynchronization. Coherence between infants and mothers during free play and the proximal watching condition, however, was still higher than in the distal watching condition. So that was the condition where both mom and infant looked at the videos on the tablet, but they were not touching. So if we're wondering whether interpersonal neurosynchrony is mainly driven by sharing perceptual input, I would say, along with really a range of other studies in the field, right now also with adults, we can say no, at least it's not the whole story. We find higher coherence during mutual engagement, face-to-face, which is arguably perceptually more different. Yeah, the more similar perceptual input in our study was certainly occurring during the watching conditions and not when infant saw the mother and the mother saw the infant. So what about physical proximity? Indeed, we see that neural synchrony is higher during the proximal watching compared to the distal watching. So when the kind of perceptual input is very similar, physical proximity seems to induce a higher level of neurosynchronization in the diet. You might have wondered why the free play interaction seemed to have a lower level of variance than the proximal watching conditions. We wondered this as well and figured it might have to do with the duration. So we split up the free play interaction into 3 epochs of 90 seconds duration, which is the same as the watching conditions. And now we see the variance looks very similar across the 5 different conditions. But yet we still see a higher level of interpersonal neurosynchronization in the free play in all of the segments compared to the watching conditions. Now, what is going on, especially in the free play interaction that is associated with a higher level of neurosynchrony? And the one thing we found was that increased neurosynchrony was related to longer durations of maternal affectionate touch, so soft stroking, loving stroking, that we coded from the videos. However, decreased neurosynchrony was actually associated with maternal stimulating touch. So this was something like tickling or poking the infant, yeah? So imagine a situation where the infant is perhaps losing interest in the interaction, trying to disengage, and then trying to get the infant back with poking or tickling. This was something that was actually associated with decreased neurosynchronization. So what qualities of the interaction are associated with higher neurosynchrony? Here we see high levels of effective touch, but lower levels of stimulating touch. Yeah, the nice thing about this study is that we looked at different levels of synchronization. So we also assessed physiological synchrony with using respiratory sinus arrhythmia, and we used cross-recurrence quantification analysis to look at the relations between the 2 time series here. And we found, first of all, no difference in the physiological synchrony between the distal watching and the proximal watching. So physical proximity itself did not seem to do the trick. However, similar to the neurosynchrony data, we see a higher level of physiological synchrony in the face-to-face free play interaction as compared to both watching conditions. Now, what in the free play interaction, zooming in again to the free play interaction and looking at the behavioral correlates of neural synchrony, physiological synchrony in this case, we see that physiological synchrony correlates negatively with positive affect of the baby, but positively with negative affect. So basically, the more the baby was unhappy and expressed negative affect in the situation, the higher the level of physiological synchrony between mother and infant. And I think that makes sense, especially taking into account or considering Sam Voss's account, the there when you need me idea. He showed in his research following mother-infant diets over the whole day and looking at physiological arousal and synchrony, that moms do not actually follow their infants or are not aligned with their infants' arousal status throughout the whole day, which would probably be very exhausting and probably not very adaptive, but they are there with their infant's arousal when the infant is distressed. Yeah, and this is probably what we see here as well. As long as the baby was happy, there was basically no need for mutual alignment on the physiological level. But when babies were distressed, this was what probably distressed the moms as well. And this could then, of course, help alert the mom and lead to co-regulatory behavior. So in this analysis, we found no correlations with touch and also no correlations with neurosynchrony. So in our task setting, physiological synchrony and neurosynchrony were not actually correlated. So is physiological synchrony established through touch? In our case, no, at least not in our really low stress setup. Touch did not promote physiological synchrony, probably because it didn't need to. Okay, so I'm going to already summarize our data and conclusion. So coming back to the initial questions for this study, is interpersonal neurosynchrony mainly driven by sharing perceptual input? Our study, as well as others, shows that that's not the whole story. There's higher neurosynchrony in conditions with active mutual engagement and actual face-to-face interactions. And this is very similar, going back to our research with preschoolers that I showed you earlier, where we found positive associations of mother-child neurosynchrony with behavioral reciprocity and verbal turn-taking. Does physical proximity promote neurosynchrony? Yes, that does seem to be the case. Being in close bodily contact promoted neurosynchrony, even in a passive viewing situation without face-to-face engagement. What qualities of the interaction are associated with higher neurosynchrony in the face-to-face interaction? We found that the duration of affective touch was positively associated with neurosynchrony, but stimulating touch was negatively associated. And we think that might have to do with stimulating touch, like poking, ticking, having at least a chance to be perceived as intrusive by the infant. So this might be something that some mothers show when their infant is actually not so much interested in the interaction or is trying to disengage. Yeah, last question, how are physiological and neurosynchrony related? Well, we found in this study that both forms of synchrony are actually increased during free play as compared to some passive watching conditions. However, they were associated with different behavioral variables, with different aspects of the interactional quality. Physiological synchrony is high, especially when infant negative affect is high. And that's, again, as I said, probably because that's when there's actually a need for it. Okay, finally, I'd like to give you some outlooks on our ongoing work. As I mentioned briefly, we are also interested in the long-term outcomes of high levels of infant caregiver neural and physiological synchrony. This is why we had these follow-up assessments, also of attachment, where we visited the caregiver infant diets in their homes and performed the attachment cue sort as one measure. We're also interested in kind of cognitive outcomes such as language and social learning. In addition, we are now looking at other aspects of interaction qualities that could promote infant caregiver neurosynchrony apart from affect synchrony and touch. So in one study by Gabriela Markova, we are looking at with the heart rate and or brain oscillations of infants entrained to maternal singing. And here we're using EEG because we're really interested in the kind of higher frequency neural oscillations that we cannot really capture with FNIRS. And also we're looking at how infants track the rhythms of infant-directed speech. And then we're also interested in the role that touch plays for prematurely born infants. So in particular, we are interested in the levels of kangaroo care provided in neonatal intensive care units, and looking at whether that also promotes neural and physiological synchrony between prematurely born infants and their caregivers. And this is something that we're pursuing in cooperation with the neonatal intensive care unit of the Vienna General Hospital. Yeah, and with this, I would like to thank my wonderful team at the University of Vienna, the Wiener Kinderstudien or Wiki Lab, especially Jin Lian, who has performed all of this research as a part of her PhD thesis. And I'd like to thank our funders, and I thank you for your attention. Very much looking forward to the discussion. Thank you.
Okay, that was an excellent talk. Huge amount of impressive work, really fascinating ideas and methods. So thank you, Stefanie, that was great. We've got questions coming in the Q&A, so I'm just going to moderate and I'll kind of read them out to you, and you can answer as we go.
Okay, perfect, thank you.
So Ian Bruce asks about the idea of the effect size and the significance here. So the quantitative changes in the wavelet transform coherence metric that you observe during cooperation are significant, but they're fairly small. Do you think that the small differences are just due to the level of sensitivity of this metric, or is it possible that we only need a small amount of neural synchrony to be sufficient in aiding in these behaviors?
Yeah, that's a great question, because, yeah, as I've mentioned, obviously the brain entrains to rhythms from the environment all the time. So it's not like we're talking about an on-off thing that's going on there. Being there in the same environment with the same stimulation, there will be a certain degree of synchronization of brain activity. But what we're continuing to see is that that's not the whole story, that it's not just some incidental, catching up or being exposed to the same environmental stimuli, but that there's something more going on. And that has to do with many different factors that are going on in the interaction. And what makes it even more complicated in our study setups, I would say, is that we're looking at fairly complex naturalistic interactions. So it's very hard, even though we are trying our best to do very detailed behavioral analysis, we will not be able to catch everything that's going on. So what we're talking about here are obviously correlations. Yeah. So to get at what causes what and to what degree or what level of, causes what effect, I think we have to go further with the methods, and stuff that we're thinking about is, or that others are also doing in other labs, is actually inducing neurosynchrony and then looking at the behavioral facts while everything else is controlled, which is pretty cool. And Giacomo Novembre is doing some work with TICS, which is something that I'm not keen on doing with kids or infants, but we can think about, targeted perceptual entrainment, for instance, which also causes, entrainment of neural oscillations. Then something that we're thinking about in collaboration with Frank Schanowski, who is also in Vienna, is trying to develop some neurofeedback approaches. So that would also give us maybe a bit more of a glimpse into causal effects if we're managing to target that or increase or decrease synchrony based on feedback. I guess those are the main methods that we have on the table right now. So right now I cannot really answer that. I would just say that's something that we're also thinking about and trying to target in future research.
Great, thank you. I want to get to a couple questions that Dorita Berger put in the chat during the talk. So this was during the third section of your talk. She asked if does emotional synchrony enter the scenario? And then another question she had that was in the same section was about whether father-infant dyads would yield similar results.
Yeah.
I think that was related to touch specifically.
That's also an interesting question. Maybe I start with the question on fathers. So maybe generally what we see in other labs research where they looked at how infants and children synchronize also with different people who are unfamiliar. It's typically found that there's also a higher degree of synchronization with caregivers, with people that the infants or children are actually familiar with. So I think it's fair to say that the quality of the relationship plays a big role in establishing interactional synchrony, or it supports that. So what I would expect is that for fathers, the level of involvement, and again, probably the identification with their own role as a father, would play a significant role there. So I'm sure I would be very confident that if the father is the primary caregiver, or on the same level as the mother, or another caregiver, then there would be synchrony as well. It's something that we haven't looked at in infants yet. It's something that I think would be very interesting and important to do. It's also something that's not, at least in Austria, it's not super easy to do, because very often, especially in the first year, it's typically the mom that stays at home with the infant. And for fathers, I guess we would have to put even more effort into the recruitment. But I think it would be worthwhile, would be very interesting. What I also found interesting in our preschooler study with fathers is that, you know, they do synchronize with their kids, and they do that in the, you know, in the interactional context. But the behavioral variables that we found to correlate in the mother-child interaction, we didn't find to correlate in the father-child interaction. Yeah, it was really this kind of self-identification, the role with the role as a father that was a predictor there. And so we well, of course, we're also talking about small samples, etc. So it's hard to make definite claims there. But it seems that fathers do synchronize with their kids, but perhaps they do that so in another way that we don't quite yet understand what's going on, like whether it's some other type of behavior reciprocity that we haven't captured with our coding system or something like that. And regarding affective synchrony, that's definitely something that's very interesting and that has been looked at in the attachment research that I mentioned, and has been shown to also relate very much to physiological synchrony, which makes a lot of sense. In not all of our studies, we have coded maternal affect. So we haven't always looked at that. Whereas infant affect, we usually code. I'm just thinking now of another study by a student who's very much interested in language development, and it's very preliminary, but she seems to find that affective synchrony in interaction between 9-month-olds and moms seems to be conducive for infants' word segmentation, but at the same age, there's a correlation with that, and we're now looking, we're actually now, well, we would be if there wasn't another lockdown right now in Austria. We would reinvite the infants at 18 months, and I hope we'll get some language outcomes on this sample. So but it's kind of an outlook, like language development outcomes are definitely super interesting to look at for us as well.
Great, thank you. Rakan asks, or says, very interesting talk, and that you mentioned earlier, you compared mothers with other children. I think this is the random control analysis without interaction. However, have you studied mothers or fathers paired with random children while interacting? And they wonder what the level of synchrony would be in this condition.
No, we haven't done that. Again, in other labs or in other studies, it's been done. Vanessa Reindl with Kerstin Konrad in their study on neurosynchrony in cooperation or competition. Also, that this manipulation. Also, they found that the actually related pairs showed higher levels of synchrony. So yeah, and Lise Piazza, I think, also in the infant study that she did. So it's fairly consistent that if the pair don't know each other, that there's lower synchrony. And this also aligns with results from adults, by the way. So romantic couples or friends, people who know each other. And some research that even goes so far that even when you look at with fMRI at the brain activity of people while they're watching a movie or listening to a narrative, there's a higher correlation of the brain activity between those who are personally closer, who are closer friends, which I think is fascinating, because that speaks to a certain degree. Well, we don't know whether, people become friends because they're more similar in how they perceive or encode information, or whether it's that people become more similar. I think there's also lots of interesting questions to answer in there, but that's just a fascinating result, and that's pretty much consistent in the adult and developmental literature.
Great. So a question from Sid Sigalowitz, getting at the kind of underlying neural measures and mechanisms and thinking about different methods you can use here. Are the neural synchrony results you present all from fNIRS, especially the very slow oscillations from 0.06 to 0.15 hertz? And with EEG, what oscillation frequencies would work here?
Yeah, so that's also a great question. So my background is actually in EEG. That's where I kind of come from. But when I started to use these hyperscanning techniques, and especially trying to, create very naturalistic interactions, I quickly ran into problems, as you might expect if you're also familiar with EEG, because you have to be very mindful of artifacts, especially when there's speech involved, when there's movement involved, and then there's limited ways to instruct babies, like none. So we are doing that, but it's very hard. So the initial studies and all of the studies that I presented today, where we have some solid results, I think those were with FNIRS, and that allows us to look at slower frequencies, as you mentioned. In the EEG, something that we have done or are currently in the process of analyzing or even writing up is looking at infants' responses to maternal speech on the one hand. So we're looking at infant-directed or adult-directed speech from the mom, also in the naturalistic context, but we're basically not even trying to measure mom's brain activity while they're being very expressive and talking with their infant, but only the infants, and that work quite well. If we have long enough interaction phases, we get enough data to make some good estimates. And there we can, of course, look at all kinds of different frequencies that are also, reflected in the speech signal. And this is super interesting. So, for instance, we can look at kind of the syllabic rate, which is something like 4 to 6 hertz. We can also look at really slower frequencies that correspond more to the prosody. And this gives us, of course, a whole different level of information into how the speech signal is basically encoded in the infant brain. So definitely different frequency bands that are very interesting there. My kind of pet favorite oscillation is definitely theta, because that also seems to be very relevant for learning and encoding of new information. So this is something that, I hope at some point we can kind of merge these different streams of research and really look at how learning happens in the interaction. But with the EEG signal, we do have problems in these naturalistic tasks.
Great, thank you. I'm going to jump ahead to out of order a little bit to a question from Fleur Bauer, because I think it's related to some of what you were saying in your response there. She's saying if she understands correctly, you did not find increases in synchronization for having the same perceptual input or for touch, but what exactly were they synchronizing to then? Was it each other's speech, or was it something else?
Yeah. So I guess that relates to the infant study. So what we did find, maybe it was just a little bit too much information, but what we did find was that the neural synchrony, or was it about the neural or the physiological synchrony? That question.
I believe the neural synchrony, but
okay.
Fleur can post another question to follow up if she'd like.
Okay, so the neurosynchrony we did find was related actually to physical proximity. So it was increased when the mom was holding the infant as compared to just sitting side by side and watching the same thing. And in that situation where we really have this, I think, neat manipulation of there's proximity and touch or no touch, what actually causes the synchrony, it's very hard to pin down, because obviously when the baby is on the mother's lap, the baby will, or both actually in the diet, will perceive each other's physiological rhythms, the breathing rhythm, the heart rhythm. And I think there are also kind of subtle, well, that's my point, my position, that touch can also act as a communicative cue or a communicative signal. So I would expect the mom to have, subtly readjusted the infant, responded to the infant moving, stroking the infant, etc. But it's very hard to code from this video. So that's what I think is happening there. And then we find yet even higher levels of synchronization during the face-to-face interaction, which I think is very interesting, because there the perceptual, well, the visual input at least is very different for the both, because they're looking at each other and not at something together. But there we did find from our behavioral coding that higher levels or higher durations of affective touch. So again, this rhythmic kind of loving stroking, that was related to higher levels of synchrony. So I guess in both conditions where we find these kind of higher or increased levels of synchronization, that touch would be the kind of the relevant communicative cue in this. And this is of course also something that I would be very curious to investigate further. So whether this then actually is also something that can be used in learning, in teaching learning interactions. Yeah, whether this could be something to kind of help get both partners aligned and then transfer information. So yeah, it's just the first clue that touch might be relevant here. But again, effective touch, yeah, not intrusive touch, not functional touch, not just any kind of being in touch, but effective touch.
Thank you. I'm going to go to a question about comparing across cultures. So this would be kind of a speculative question, but Daphne Maurer is asking, how do you think they might generalize these results outside of the weird population, Western societies, where patterns of mother-infant interaction might differ? So their affective touch might be less common, or that kind of thing.
Yeah, that's so interesting, because we know from behavioral studies and observations that actually being carried and being engaged in really kind of physical rhythmic interactions is very much a part of caregiving in societies other than the kind of Western industrial context that we've been looking at so far. So yeah, it's hard to know. It's hard to know, obviously, without having done the research there yet, but just from knowing this literature on how caregivers tend to interact with their infants in other cultures, I would take a wild guess that rhythm and synchrony plays a role there as well and is used there as well. Yeah, infants are carried a lot more. So even if there's, I don't know, maybe not so much effective stroking or we know there's less face-to-face interaction, that's for sure. But being carried a lot, being rocked a lot, having all these physical, really activities and games that are very typical to observe in these contexts, I would guess served similar functions in the end, like aligning brain activities, promoting information transfer, promoting coordination and cooperation. So this is super interesting. Also, I think in the thinking about different cultures where there's not necessarily only one primary caregiver or kind of the small family context, but interactions with peers play such a, like just a little bit older kids or really same age kids. This is also something that hasn't been, we're just, starting to understand a bit better what rhythms and synchrony, what role they play also in peer interactions. This would be also super interesting to look at in across different cultures. I totally agree.
Great, thanks. So we've got about 6 minutes left, and these are really excellent questions, so we'll get through as many as we can. Laura Sorelli is asking, going back to the response to the previous question even, what's going on with intrusive touch? So do you think that intrusive touch is causing the decrease in neural synchrony, or is it a strategy perhaps when there is a disconnect happening and neural synchrony is less? Would you be able to look at the level of neural synchrony right before the poke or the tickle to address this?
Yeah, that's an excellent point. It gets at kind of the this is something that again something of a construction side that we have to work on or dive into, kind of looking at really the temporal dynamics and what causes what, because this is definitely something that we have in the information that we could get from these time series, and that we have yet to do and to understand better. So my intuition would actually be that this is something that that moms in the study, like also looking at the videos, that moms probably did in order to trying to re-engage the infant. So if the infant, yeah, again, it's hard to say what causes what, but my intuition would be that in interactions that weren't so smooth and where the infant was perhaps trying to disengage, and moms, of course, were in the lab context, so wanted it to work, that this is something that they might have tried poking and picking and tickling, just get the infant's attention and get it going again. But at least in this context, in this age group, in this context that we had, it seems to not have been so functional, probably. So yeah, but definitely on the to-do list.
Great. There's a couple questions here that are going back to this issue of like what's going on in the environment and the behavior that might be driving some of, or the synchrony measure. So Erica Flayton's asking if you controlled for the factor of gaze. And then Haley Cragness was asking again about speech, whether that might be driving some of this, that was there more speech, for example, in the free play condition compared to the other ones?
Yes, that's a good point. So we definitely look at, so yeah, both actually, we code gaze and we code speech. So perhaps going back to the kind of preschool work, their speech did not explain the differences between the conditions, and we actually had some speech also where they played individually. Then in the infant study, we did control for gaze. So we did not have, for instance, we did not have differential looking at the screen and the joint watching and the distal watching condition, because this was also something that we considered. Could it be that they're just more attentive to the screen when they're held as compared to when they're alone? And this could, because the mom is always looking at the screen because we instructed them, and this could lead to high alarm, but that was not the case. Yeah, and then in the free play interaction, I would have to look at the data, but as far as I remember, there we didn't actually find that speech was related to the neurosynchrony, but I would have to look it up. I'm sorry. I think what we also looked at, but this is work in progress. I'm sorry. It's just these data sets are very rich and it's hard to kind of have everything in mind all the time. I'm sorry. But what we're also looking at now is whether the reciprocity, the turn-taking, the vocal turn-taking in the free play interaction, whether that relates to neurosynchrony. So yeah, so the maternal speech alone, the speech signal was not really kind of explaining the variance in the neurosynchrony there. But now we're looking at, also again as a kind of measure of mutual engagement and reciprocity, whether the turn-taking quality in the mother-infant interaction, whether that relates to neurosynchrony, as it did in the preschoolers, but we're not so sure about the infants there. So yeah, work in progress.
Great, thank you. So we've just got 1 minute left, so probably the last question here. Michaela Santangelo is asking about comparing mothers and fathers' neural synchrony, wondering about whether the results could be affected by age or sex of the children, or about whether you can control these variables. So a mother or father could exhibit different kinds of neural synchrony if their child is a boy or a girl, or if it's a younger or older child, that kind of thing.
Yeah. We definitely looked at that, and yeah, I again would have to look it up to be really sure, but I'm pretty sure that the sex of the child did not have a significant effect. Yeah, because yeah, obviously you could, you know, there could be something. Yeah, it's unlikely, but there could be something physiological going on having to do with, I don't know, hormones, brain structure, that, you know, sex-matched diets express higher levels of synchrony. But I have to be honest, even in adults, I'm not aware of that. And this is something that is typically controlled, or, you know, at least it's in some way acknowledged in the analysis. So yeah, no, to the best of my knowledge, I don't think that has played a role in any of the research that I'm aware of.
Okay, thank you. I'm so sorry to have to cut it off here. This is such a great discussion. So many good questions we still haven't gotten to. So I just suggest to people that if you're able to join the lunch sessions, perhaps you'll have a chance to talk to Stefanie there or during the panel discussion at the end. So let's try to keep this great interaction going. So thank you so much, Stefanie. Wonderful talk, wonderful discussion. Appreciate it.
Thank you so much.